हेलो। you for coming. Thank you for coming in.
Say
Thank you for coming in for the colloquium.
Oh, yeah.
No, it just failed yesterday. They don't have an extra. How is this possible? The institute doesn't have an extra projector. I sent a mail directly. This is the older one.
Yeah,
Okay, I think there is some sort of a quorum. I will let people come in. So I would like to welcome one and all for this edition of the Wednesday colloquium. And we have a special colloquium today. We have Professor Shyamalava Mazumdar, who has been in the institute for a long time, almost 35 years, close to 35 years, and has been has seen the institute evolve from what he joined as a young student to now as he is sort of coming out of the institute, giving us a goodbye. This has been a long journey for him, and this particular colloquium will try to capture some of the excitement and the fun he had over the years, looking at the research problem, of course, that he's going to talk about. But apart from that, I would like to, at least since I am the colloquium coordinator, it gives me a chance to introduce him. So I will sort of give you a little bit of a short introduction to Professor Shyamalava Mazumdar, especially for people, for students who are going to listen to him for the first time, either way through the YouTube, Zoom, or currently as you are sitting in this auditorium. So Professor Shyamalava Mazumdar actually did his bachelor's in chemistry from Vivekananda Mahavidyalaya, which is part of University of Bardhaman in West Bengal. And then he sort of completed his master's with specialization in inorganic chemistry from University of Bardhaman before coming here as a young PhD student, just like many of you who have joined through the entrance exam. And he sort of joined the institute in 1984. That's when he started his work in the sort of things that he carried on as a traditional, looking at very complicated molecules, some of the structures that are shown here. But he looked at Hemes, which are these class of molecules, using both electronic structures of Hemes using NMR spectroscopy. And this is where the uniqueness of Shyamalava's PhD thesis is. He sort of was one of the few people who at the very early stage used NMR spectroscopy to look at heme structures. Okay. And a very unique case also in the point that he published single author papers during his PhD, although he had a mentor, Professor Shamarosh Mitra, who was a very well-known inorganic chemist, bioinorganic chemist of his generation. But Shyamalava actually sort of went solo in a couple of the papers, and that's pretty amazing at that time. And as an experimentalist, okay, I do not debate. There are other departments in TIFR where single author papers are not very uncommon as a theoretician. Then actually he went to do a short postdoc with Professor Alan Hill in University of Oxford, after which he came back and he joined as a reader and then went to an associate professor to a professor in very short span of time, leading the work in bioinorganic chemistry in India and the world, and putting India on the map in sort of looking at metalloproteins, which he's going to talk about here. Okay. He is, of course, because of his wonderful work, he has been rewarded at multiple levels. Of course, he was one of the recipients of the original Geeta Udgaonkar Award for the best PhD thesis in TIFR. But apart from as a student, he was a young, he won all the young scientist medals that the, Indian National Science Academy could provide, different kinds of academies could provide. And also from the Chemical Research Society of India, he has been, he won a medal, a young, young scientist medal, the bronze medal, as we call it. One of the critical parts that I would like to say is my personal association with Professor Shyamalava Mazumdar. He actually visited Princeton University when I was a PhD student. He was visiting at that time Professor George McClendon's lab, who specialized in looking at electron transfer in proteins, in metalloproteins. And that is where my first sort of, after my VSRP, interacted again with Professor Mazumdar. And that's where I think the energy and the enthusiasm of coming back to India came for me as well, talking about science in India as well. So I thank you for that. And with that, I would like you to do the colloquium for all of us, which is Journey from Hemoglobin to Cytochrome P450, a story of different avatars of the heme.
Thank you, Jyotisman. He has almost everything he has told, so I only have to show the slides. So actually, I think everybody knows about heme. I thought that I should also identify myself as a chemist, so show some chemical structures and also something which everybody knows. Okay, so this molecule called heme is very common because everybody has hemoglobin and blood. So hemoglobin, heme is actually a iron complex of a organic molecule called ligand, which is called porphyrin. And this porphyrin has, as you can see, that there are 4 nitrogens at the center, and out of that 2 nitrogens have hydrogens. And when you replace these 2 hydrogens with metal ion, and then you get, do I have to do this? Yeah. So you get iron complex, okay? And this heme actually forms the core of hemoglobin. And it's not only that, that heme center is present in many, many other proteins. And it is present even in bacteria, okay? And without heme, I think all the aerobic life would not have existed. And there are varieties of functions the heme center does. So I have been really very fascinated by just looking at and learning this one molecule. There are so many molecules chemists deal with. This one molecule nature has chosen to carry out multiple different type of functions. So what is that, as a chemist, what is that you look at, which makes it doing different type of functions? As a matter of fact, this porphyrin ring is something which nature's choice. And some slight modifications in the side around these, or in some of these double bonds and all that, nature produces different type of molecules which are used for different functions. For example, chlorophyll, which is one of the most important molecule for the survival of the life on Earth, actually is a magnesium complex of a ligand which is derived from porphyrin. So there are lots of things which are there, and I have spent more than 3 decades, and almost half of my research activities have been directed towards understanding how this heme, the variations of the heme's function and properties happen in biological system. So this is the outline of my talk. I will talk to you about heme, introduce you about some of the different type of hemes. There are some natural hemes, and also there are synthetic hemes or synthetic porphyrin. People make it, which has many other commercial applications and photophysical applications and things of that type. And various types of functions of heme. I'll spend a little bit time on hemoglobin and its younger brother, myoglobin, which are involved in oxygen transport and storage in us. Then I'll talk to you about electron transfer, which is a quite different function of heme. And there is a concept of what is called reduction potential, redox potential. I will move on to talk to you about some of those enzymes which has heme. Enzymes are biological catalysts. For example, some of the enzymes which are, they call it peroxidases, they are the antioxidant enzymes, which are found in many of the green vegetables and radish and things of that type. And there is another enzyme which I have been spending last more than a decade, is an enzyme which is called Cytochrome P450, which is involved in synthesis of steroid hormones in our body and also do drug metabolism. And we have found there are various possible applications of those enzymes and their analogs. And that is what takes me to modify the heme environment and how we can do protein engineering to actually give different type of function to the heme center. And there are some sort of unnatural functions that also can be carried out by doing this type of, playing God on the protein. So let me just tell you what is heme. The heme is what they call it prosthetic group. That's non-amino acid component in many proteins, and those proteins are called heme proteins. And the deficiency of iron in blood is actually deficiency of heme. That is what is found. And although the presence of heme is known since 18th century or even before that, the first time the structure of heme was derived by Hans Fischer, and he got Nobel Prize for that. And when he did it in a very synthetic way, those days extra structure was not available, and he found many isomers, many side chains, components around this ring, and the 9th isomer of his synthetic products matched exactly with the species which is found from the blood, and that's why he gave a name called protoporphyrin. So heme is also called protoporphyrin 9 complex of iron. So that's the common thing. And because of presence of this conjugated single bond double bond structure, having the ring is a highly aromatic molecule, the porphyrin ring, as well as the metal complex of that. And it has, because of that very high intense color, it has a visible light absorption. The porphyrin ring also emits. Iron complex does not have emission, of course. So understanding of the properties of this heme in biological system basically boils down to understand the electronic property and the chemical property, reactivity of this metal center inside the protein. And there are so many spectroscopic techniques, starting from when I was a student. I used to study Mazumdar spectroscopy of iron in heme. We did NMR, that also can be done. And nowadays there are even mass spectrometry and many other techniques. Sort of sky is the limit. Anything can be used. These are the type of hemes. This is the core and having different type of what they call substituents. And iron generally, typical inorganic chemistry tells iron can bind actually to 6 ligands. Out of that, 4 centers are occupied by the porphyrin ring, and there can be 2 other external donor centers connected to the iron. And that's how sometimes it gets anchored to the protein through some amino acid. There are naturally found, this is the heme, where you can see there are 2 side chains having carboxylic acid group. They are the acidic groups. And this side there is a vinyl group and methyl group at the top. They make it more non-polar, and this side is polar. So there is an orientation effect in the structure. And there are slight variations, like for example, this is called heme B. You can see heme C, heme A, and heme D. They're all found in different proteins depending on the type of function. But these variations are very minor. They don't affect the main properties. You can actually replace one from another, and it has this type of variations has been observed. There are synthetic hemes also, but of course, this code, this tetrapyrrole code, which is called porphyrin, till today it is not been synthesized. Nobody has been able to make it. Always there are some side chains has to be there. So these are the challenges of synthetic chemistry, which I am not going to talk to you about. These are the spectral features, as you can see. Because of this presence of aromatic features conjugated as single bond, double bond, they have a very high intense peaks. One peak around 400 nanometer, that is around in the green region, is a very intense absorption it has. And that's why most of these metal porphyrins, even the blood hemoglobin, has a red color because it absorbs green color.
Very easy question. Is it synthesized inside the body?
Yes. I am not talking about that, but maybe at the end I will tell you how exactly it's synthesized. Now it is known. There are 2 mechanisms. One is in case of plants, and other is for members. There are people have made different other metal porphyrins, of course, synthetic porphyrins, and they are used for as nonlinear optical studies, They are used for photodynamic therapies and many other things. Okay, so they have commercial applications. Okay, and many other applications which I do not know. They also have, because of high aromatic property, they have tendency to form aggregates which can be used for making batteries and all sorts of things. But my today's topic is to talk about heme, which is iron porphyrin complex, and it carries out different type of diverse chemical functions depending on the nature of the protein environment in which it is sitting. For example, oxygen binding, as I mentioned to you, hydrogen peroxide or superoxide degradation, which are antioxidants, and also the conversion of oxygen to water. That's the terminal stage of respiration. That's also being carried out by the heme inside a protein. And the last one, which I mentioned to you, is the P450 function, where monooxygenation of different type of molecule, including steroids or drugs and things of that type, happens. So this diversity of the function of the heme arises due to interaction of the amino acids and other things which are present around the heme in the three-dimensional structure of the protein. So the heme actually is trapped inside the protein polymer structure, and that molecular level interactions, both bonding interaction as well as non-bonding interaction, which chemists talk about, are the origin of this type of variations. So these are the different type of activities. For example, oxygen transport and storage is done by a class of protein which are called globules, like hemoglobin, myoglobin. There are transport proteins for nitric oxide. Nitric oxide is a neurotransmitter, and there are proteins which actually transport it. Carbon monoxide also is another neurotransmitter. There are proteins which can transport that from one place to another, like nitroporine is one of them. That also has a heme.
We have shown this
iron valency as 3+, or something like that. Is it 3+ always? Because
No. That's the first thing I'll talk to you about.
Okay, so I was curious to know.
I'm just introducing you. You can have 3+, 2+, 4+ also.
Yeah, because the absorption spectrum also, whatever you have shown, there also the peaks are almost at the same position.
They don't depend on the metal. As I mentioned to you, the porphyrin ring is highly aromatic, and that's the one which gives color. So this metal center only just a little bit shifting. And there are catalysis by heme-containing proteins, which are also there are different type of enzymes which are involved. But of course, in most of the vertebrates, hemoglobin is the major thing, which gives the red color of the blood. And for example, in human body, almost 750 grams of hemoglobin, which is the major source of iron in our body. But there are iron playing much more vital roles in our body, which I will tell to you later. So let's start with myoglobin. I think myoglobin is the first molecule, first protein whose crystal structure was done by X-ray crystallography, and by John Kendrew, and Max Perus did the crystal structure of hemoglobin by X-ray crystallography, and because of that, they got Nobel Prize in 1962, almost when I was born. Okay, and that was the beginning of connecting structure to the function of the metal center in biological system. Myoglobin structure is now known. Okay, and as you can see, I don't want to go into detail of this spaghetti structure. As you know, it's the backbone the protein, and the amino acids are not shown here. I show you the heme molecule, which is anchored inside the protein. Okay, and there are two amino acids. This is one amino acid which is called a histidine. It has a nitrogen, and that nitrogen is actually coordinated to the metal, which is called proximal histidine. There is another histidine molecule which is quite far, and it's not coordinated actually, but it is called distal histidine. But biologically, this also has a function. If you remove that, myoglobin will not bind oxygen. Similarly, the hemoglobin also has hemes, but hemoglobin, unlike myoglobin, hemoglobin has 4 subunits. There are 2 alpha and 2 beta, and they are slightly different in molecular weight, but they also have the heme having a proximal histidine. That means one histidine is ligated, another histidine is a little far away, and not directly binding to the metal, but they have very important function. Now, what is the function? Function of hemoglobin is oxygen binding. That's what is the main thing. Myoglobin also binds oxygen. Hemoglobin's job is to bind oxygen in the lung, and then it takes to go to the interior of the body where there is no oxygen, and to the cells or muscles, and there it releases oxygen reversibly. And in muscles, the protein which is there is myoglobin that accepts the oxygen. So it's a reversible process, very, very smooth, almost extremely fast process. So the iron porphyrin in hemoglobin as well as in myoglobin, in absence of oxygen, they call it deoxyhemoglobin or deoxymyoglobin. And when oxygen binds, it binds to the 6th position. As you can see, in absence of oxygen, the iron has 5 coordination. This one of them is histidine, and when oxygen binds, the 6th coordination is occupied by oxygen molecule. And the point which you are telling, in case of hemoglobin myoglobin, it is the iron in reduced form, iron 2+. If you become iron 3+, it will not bind oxygen. One more interesting thing, cyanide does not bind to iron 2+. So there is a wrong notion that cyanide binds to hemoglobin and that's why cyanide is poisonous. That's not correct. Cyanide does not bind to hemoglobin. It binds somewhere else, which is much more important. And because of that, although bacteria or ants don't have hemoglobin, but they will also get killed by cyanide. Hemoglobin and myoglobin, both of them has a very strong affinity to carbon monoxide. And because of that, in Indian villages, in winter especially, there are a lot of people have this die because of carbon monoxide poisoning. So carbon monoxide actually has a much stronger binding affinity for the ferrous heme molecule. Okay, if you have ferrous heme just bound to a histidine, the carbon monoxide binding affinity is 25,000 times more than that for oxygen. But nature has designed in a way that distal histidine, which just sits above this, as you can see, I have shown you in the schematically here, so that the carbon monoxide cannot bind the way it likes to bind. So carbon monoxide binding always likes to have a straight binding. Okay, and because of that, the binding affinity of carbon monoxide for hemoglobin, heme in hemoglobin as well as myoglobin, decreases. And even then, though it decreases, it's almost 200 times more than that of oxygen. So if your concentration of carbon monoxide is 200 times less than that of oxygen in the air, that itself is good enough to cause a carbon monoxide poisoning. So the important point is that hemoglobin and myoglobin has iron as a ferrous iron in functioning. So functional form of heme is ferrous iron. In fact, in our body, there is a molecule called glutathione, which is continuously checking and reducing heme of hemoglobin to making it reduced, is maintaining it in the reduced form. Because if it becomes oxidized, that becomes poisonous. So it has to be in the reduced form. So the question is that what makes, as a primitive question, very what to call simple question, that why does iron becomes iron 2 in hemoglobin myoglobin? If you take out the heme, you can very easily remove the heme from protein. That always have a iron 3+, which is called hemine. Okay, hemine is very stable as a thing. So what makes it stable as a ferrous inside the protein? So this was my first independent work, I should say, when I was a very young boy. I just took soap myself, and we encapsulated heme inside the soap, and we found that there are spectroscopic evidences. Of course, we did a lot of NMR studies on that, having these spectral features which are exactly matching with the ferrous form of heme. That means the ferrous form is stabilized by the environment which is created by the micelle, which is a hydrophobic environment, because the organic molecules of the micelle, organic side chains, they actually remove the water access from the metal center. So possibly the protein in hemoglobin, myoglobin, they do similar type of things. Okay, they remove the water from the proximity of the metal center. We did detailed NMR studies. Those days, my thesis was actually on NMR to find out where exactly the heme sits inside the micellar cavity. And we showed that actually it sits in a sideways, in a very specific orientation. And later on, we did quite a lot of fluorescence dynamic studies also. Subsequently, we looked at how exactly now surfactant can provide interaction, which is called hydrophobic interaction, which is similar to that of protein. That's the sort of a very simple picture. So if we take protein also has this hydrophobic interaction. So if we interact the protein with the surfactant, what happens? So we see that hemoglobin, both hemoglobin as well as the, we also worked on cross-linked hemoglobin. And when we add very, very small concentrations of the surfactant, SDS or, different type of surfactant micelles, slowly the heme comes out. And that process is very slow, but it does come out. And the spectral features changes. My first student, Tapan Das, was just 3 years younger to me, actually. So he actually, those days we could publish in ChemCom by doing this 1-week experiment where we could show that when nitric oxide is coordinated to the heme, this release of heme from hemoglobin or myoglobin is much faster. And this was in 1990s. And that was the time guanylate cyclase was discovered, and this nitric oxide, how exactly it functions. And we could mimic the function of guanylate cyclase that when NO is bound, then only the heme comes out of hemoglobin in case of guanylate cyclase activation. Anyway, let me just deviate a little bit and talk to you about a technique which actually had been my other than NMR and fluorescence, my major source of eye to view things is electrospray ionization mass spectrometry. This is a mass spec, normal mass spec, as you know, that we get the peak for M by Z. But for a protein, for the same protein, even if you have 100% pure protein, you don't see only one single peak. You get multiple peaks. Okay, these are called multiple charge state. You get a charge state distribution. Each peak corresponds to certain charge state. That means the surface charge are different. Okay, and overall molecular weight is, let's say, for example, cytochrome C is 12,000 odd. Okay, and it shows multiple charge states. So if you take in different pH, the charge state distribution changes, but its molecular weight remains same. That means the protein undergoes some sort of change in the structure, so that the charges on the surface changes, but the protein remains same, molecular weight remains same. Cytochrome C has a large number of positively charged amino acids, so it is a positive protein. In spite of that, we found that it also shows negative mass spectrometry, so negative charge also shown. That means there is some mechanism which is there, which stabilizes different type of charges on this molecule during the electrospray mass spectrometric technique. So, although this is a positive thing. Now, this was around that time, this paper came out in biochemistry by Lars Konerman, where he showed that if you take myoglobin, where you have taken out the heme from that, which is called apomyoglobin, you get a charge state distribution like this. There are lots of peaks. Each of them have unique charge state, and you get a distribution of charges. So this distribution of charge state corresponds to a structure of apomyoglobin. Now what they did, they added heme to it from outside and looked at the mass spec with time. As you can see that after some time slowly, there are new peaks start coming. After a few seconds, you can see the distinct peaks which are marked as a H, means hollow protein. Hollow protein means bound to metal center. And after 15 hours, you only see the metal bound peaks and no peak from apo. That means when you have the apoprotein and add the heme, slowly the heme goes and binds in the right place. So it's sort of a recognizes the heme where it is supposed to go. There is a interaction.
The addition was in the solution itself, right? And then sprayed.
Yeah,
in the
it's a
vessel.
stop flow
It's
mixed
stop-
with that. Okay, but it's sort of you can see the diagram shows that. Okay, so that means there is a sort of a recognition of this thing, and second thing is that when heme binds, the structure also change because the charge state distributions are different. Because as I mentioned to you, the charge state distribution is a measure of the structure. So as the heme binds, the structure of the protein also changes. So we looked at this, what is this origin of this charge state distribution? And those days, the conventional charge state distribution analysis people used to do using this simple spherical charge particle model, which is called Rayleigh limiting charge theory, where you assume that it's a sphere and has some charge, and depending on the size of the sphere, what is the maximum charge it can have. That's the limiting charge. So using that theory, you expect for a protein like cytochrome C, whose molecular weight is known and things of that type, it should have a maximum charge of +9. Okay, but what we find that you can have multiple charges, and not only that, you can have charge more than 9 as well as less than 9. So my this Halan Prakash in my lab, he actually looked at the structure of the protein. He actually studied possibly almost 50 proteins, not only the heme protein, but other proteins whose crystal structures are known, and mapped different type of charged amino acids and found there are 4 different interactions are possible. If you have a positively charged amino acid on the surface, and if nearby there is a negatively charged amino acid, they can actually form a Zwitter ion. Okay, but on the other hand, you can have separate positively charged amino acid which has nothing nearby, or separately there is a negatively charged amino acid where nothing. So this type of classification, and if we have this type of interactions, then although they are possible to, in principle, individually they can have charge, but resulting to 0 charge. They will not be able to carry any charge. So that way, on the other hand, if there are surface exposed free positive residues, like then they will have positive charge, or if you have a free negative residue, that will have a minus charge. And whole structure can be analyzed. Now you can actually directly analyze the 3-dimensional structure and tell how many charges, what are the maximum charges possible. And based on that, the maximum charges were calculated, and this black dots are the ones which are calculated based on this model. And the red dots are the ones which are experimentally reported, not only by us, but others also. And this blue one is calculated just based on what I mentioned to you, Rayleigh theory. So Rayleigh theory does not really match, but our theory, we could show that actually it matches it. And we were the first to directly correlate the crystal structure of a protein or the folded structure of the protein to its mass spectral feature. And this is a beginning of using mass spectrometry to probe the structure of a protein. Now, where is it coming in case of heme? So if we look at the heme structure or in myoglobin, cytochrome C has a different type of heme. It doesn't come out. But in case of myoglobin, you can see here, there is a charge state distribution, which Konerman also has seen. Now, if we perturb the structure a little bit by adding alcohol or something like that, the protein structure gets a little bit weakened, and heme reversibly comes out. And we see that you can have this small peaks, and they slowly grow up, having similar type of distribution. So you can actually have a folded structure of the protein, okay, where heme is present or absent. So it can maintain a, which is possibly at some stage there was a lot of debate and dispute about what is called molten globule structure. And it might be of similar type of things, that having the structure intact with and without heme. But definitely binding of the heme to the protein adds to the stability of the protein structure that we have seen later on.
clarification.
Just a clarification on this data, actually. The x-axis here is showing different proteins, is it?
Yeah, these are different proteins.
These are different proteins.
These are all different
Heme
proteins.
Right? And
Almost 50 proteins history. That's what I'm telling.
Right.
And they have different structures. And for each different structures, we are analyzing what is the charge state.
so the structures were known for all of these 50
All of them,
proteins in that case. Yeah.
from the protein data.
So I have a very stupid question. So you said that there is some kind of theoretical that RLCT or whatever that blue thing, and then you had your own model that you have done, and then the data is fitting. So what exactly was done at the model level? Like you start with the known knowledge, and what did you do?
So RLCT is a model which just takes the molecular weight of the protein and assumes that it is a spherical structure, okay? And based on that, it calculates what should be the maximum. So, it is known that, if you have a charged sphere, what will be the maximum charge it can carry? That's the Rayleigh limit, and it gives that. And what we have done, we have taken the crystal structure of the protein, which is independently determined or reported, and looked at the amino acids and found how many are charged residues and how they are poised, so that those which are surface exposed, but they are not interacting with any other opposite charge centers, they are the ones which are counted, and that's how we calculate. So this is what is the model which was performed. This is sort of a what to call structure-based things. So I just give you some bit of contributions on the hemoglobin, myoglobin. Let me just tell you about the electron transfer proteins. Electron transfer is a very, very important function, and in fact, most of the metabolic processes, living system, is somewhere or other electron transfer to a large extent. I don't want to tell you about what is redox potential, but redox potential is the connected to the free energy of this electron transfer process driving force, and more positive the value of redox potential means more oxidizing that species is. That means it will take out the electron from whoever is available, and then less or more negative means it's more reducing. Okay, for example, but as you can see that all of these proteins which I mentioned, HRP is horseradish peroxidase, myoglobin I mentioned to you, and cytochrome C is another heme containing protein. All of them, it is the heme center which has the iron, and we are looking at the redox potential of them, and they have a various different type of redox potentials. The table can be made a little bigger. I'll just show you in the next slide. Now, Krishnananda actually had shown that again this micellar environment, okay, and he used micelles. He also used different micelles actually. This is the plot. I don't want to go into detail with the different ligands, okay, to the heme. Here there is a heme, is a different heme, slightly different, and actually calculated different components. For example, axial ligand, how much is the redox potential variation can be. Hydrophobicity means micellar environment, how much is the redox potential variation can actually lead to. Some sort of a qualitative scale of this he could come up with, okay. And in nature, actually, we see, for example, these are all cytochromes, and their function in biological system is to do electron transfer in different species, having very similar type of geometry. You can see that heme having two axial ligands, but their redox potentials vary over a wide range. And you can actually have heme proteins having the heme with redox potentials vary from plus 400 millivolt. It's not very small amount, okay, to minus 400 millivolt. 0.4 volt is not a very small thing. So this type of variation of the same molecule is achieved by varying the environment around the protein. So how does it achieve? One of the ways is by changing the axial ligands that in these cases, as you can see, are more or less same. So there might be something which is beyond the what is called first coordination sphere. That is, it's not only the metal center directly interacting and binding to something, and that's why affecting the redox potential, but something more than that. So this was the time I actually went to Princeton and met George McClendon and also JD. This was just after 9/11, actually. And he had a protein called Cytochrome B562. This is a very nice looking protein, okay, having this helices, and the heme is sitting inside and extremely stable. And this is found in E. coli bacteria. Until today, nobody knows what is its actual biological function, but it's very stable. And then and crystal structure was known. So what was done is taking these and identifying few amino acids near the heme, okay? And that was the time when, I think before that, Michael Smith made this discovery of PCR and got Nobel Prize, and it was very hot thing. No, I think this was much later than that. It was in 2001. And in 90s, I think it was 95. So what we did, we identified 4 amino acids, 2 hydrophobic, non-polar, and 2 polar amino acids. And then by doing genetic engineering of the amino acid, of the gene code corresponding to those amino acids, we randomized them. That means for each amino acid, we put all possible, 20 different amino acids. So if we simultaneously change 2 of them, we actually get 4 variations. Okay, and taking 400, 20 by 20, 400 different type of proteins having variations only in those 2 amino acids. And then we determine the redox potential of them. Okay, and when you do that, ignore these things, how they are determined and all that. And redox potential is plotted here. And you see that just varying these two hydrophobic amino acids, you could actually, vary over 120 millivolt. And they are not interacting with the metal center. So they are, very weak interactions. And nature actually uses this type of interactions to tune the redox potentials. So it has been shown, for example, in respiratory systems and others, same metal, electron is moving from one center to another center. And that's how it is done. Okay. Similarly, we looked at the other 2 amino acids, and you could get even more wider variations in the redox potential. So we could demonstrate that the redox potential of the metal center, which is actually, if you look at inorganic chemistry textbook, you will see that, different at the end of the book, you will see in the appendix, redox potential of different metal ions. Okay. But now we understand that, it's not something which is invariant. It can depend on various factors, not only the, its environment, and its sort of non-bonding interactions also has a lot of effect on them. So let me just now, maybe I have another 15, 20 minutes. Huh? 15 minutes. So let me talk to you about enzymes, heme-containing enzymes. That's something which is the largest area. And what I feel that, these enzymes are basically catalysts, and they have evolved over, billions of years since the life started. Is the enzymes which have been, doing reactions which are important for the life. And this is true for all type of enzymes. And as organisms become better and better, higher and higher organisms, they have much more sophisticated and more optimized function, okay, and regulated functions. So Frances Arnold got Nobel Prize in 2018 for discovering a technique called directed evolution. So what she did, she took the protein of choice and made the modifications in the laboratory by randomizing the protein sequences, amino acid sequences, by genetic engineering. And then from that, screening the protein which is having the best function. Nature possibly has been taking thousands of years or maybe millions of years to do that same job, which she is able to do it in laboratory.
Sorry, Shyamalava, there is a question, long-standing question from the Zoom. Shrishti, Shrishti is one of our students. Shrishti, could you ask the question? Shrishti, can you hear us?
Yeah. Am I audible?
Yes, you're audible.
Yeah. So so during the talk you have discussed about like how we can calculate the surface floating charges. So like which experimental technique you have used to calculate like the total charge which is on the surface of the protein?
The surface charges, how do we quantify it in terms of a technique?
Yeah, so you have the crystal structure as the input.
Okay.
You can just write your own routine to calculate the charged amino acids and assign one charge to each amino acid, and then look at the distance between two amino acids, nearby interactions, and find out whether they can actually form salt bridge or hydrogen bonding interaction. That's how we could calculate total charge.
Okay, so you calculated like manually. There was no experimental technique used.
No. It's not experimental. This completely computed.
Okay.
And experimentally, you get it in the by mass spec.
Okay. Yeah. Okay. Thank you.
So Francis Arnold first showed that you can use directed evolution, which is a sort of a random mutagenesis, and finally you get the protein which has a different function. Okay. So we thought that there can be a better and more easier technique. You can take a protein. Good thing is that if you have a heme protein, you can actually probe them very easily because heme has a color. Okay, and various other spectroscopic tools can be used to look at the protein's catalytic center. Okay, inside the protein, and the reactant, which are called substrate, they actually bind close to the metal center. Okay, so you can actually have some enzymes which has very broad spectrum substrates, so they can be utilized. That is one way. Other way is that you can actually do some sort of molecular modeling and molecular dynamics simulations to design the active site of the protein by doing what is called rational design, which I will just briefly outline. Before that, let me just tell you about peroxidase enzyme. This is another very, very important enzyme, which is called antioxidant enzyme, which is first found in radish, which is the horseradish peroxidase. It's commercially used as a component in ELISA kits, okay, because it's extremely sensitive to detect hydrogen peroxide, okay. And it has a catalytic cycle where it forms, as you can see, Carlo, that it has a resting state is iron is 3+, okay. And in the intermediate, it forms a iron 4 with a free radical on the porphyrin ring. And in the laboratory, you can actually add hydrogen peroxide. You can actually spectroscopically see this. That means it's stable. The protein has a mechanism by which it can actually produce and stabilize this free radical and also iron 4, which is not known in as a chemistry thing. And then you have another intermediate where you have iron 4 species. It's a ferryl oxygen. So these are very short, highly reactive things. And what people have done is to understand whether we can use a smaller protein, very well known, for example, myoglobin. This is the myoglobin structure, and this is the horseradish peroxidase structure. Horseradish peroxidase has a molecular weight of 42,000, whereas myoglobin is 17,000. So you can tell that myoglobin is the model of horseradish peroxidase. So Mike Smith, who got Nobel Prize for discovering PCR, he looked at the structure and found that in both of these two proteins, the heme center has one axial ligand is a histidine. You can see there is a histidine here, nitrogen is bound. And also both of them has another histidine 64 here. Here there is a histidine 42, which are distal. They are not directly coordinated. But only difference is that here the distance is 4.3 angstrom, here it is 5.6 angstrom. And that distance difference is the one which makes myoglobin binding oxygen as an oxygen storage protein. On the other hand, HRP does not bind oxygen. It actually stabilizes iron 3 form. In case of myoglobin, it's iron 2. And HRP actually reacts with hydrogen peroxide and it forms iron 4 species. So what he did, he just converted this histidine 64 of myoglobin into a leucine, which is not a coordinating ligand. And there is a phenylalanine, which is, this pointer is not working now. Phenylalanine, which is, no, sorry, I think that's okay. Phenylalanine, which is here, around 5.3 angstrom away, okay, that's made into histidine. And he showed that, this mutant of myoglobin had catalytic activity, not as much as that of peroxidase, but it has increased. Okay, so he is sort of mimicking HRP activity by modifying the myoglobin. So he's trying to create active center around the metal center, around the heme, very similar, so that you can get the similar type of function. So this is structure connected to the function of the heme. Okay, this is sort of a beginning of this area that you can use smaller protein as model for much more complicated protein. And it can be, for example, there are certain proteins or enzymes which produce extremely commercially important chemicals, but those proteins or enzymes are not very stable, or they're membrane bound or something like that. So can you use, smaller, well-known protein and modify them suitably so that you can use them for making these high-value chemicals for industrial use? So that was the beginning of this idea came up. So we also modified cytochrome C and showed that cytochrome C modified, where axial ligand is changed, could have the peroxidase type of activity. Meanwhile, Topham, in this connection, he did time resolved fluorescence, where he looked at HRP and HRP without the heme, and showed that in absence of the heme, the HRP is a single tryptophan protein, and looked at the tryptophan dynamics, showed that actually the protein has multiple substrates. So the conformational dynamics becomes more if heme is not there and when heme is present. So heme interaction with the protein even present in HRP also. Although
so that was my question. I did not ask it because you were running through this, because the distance criteria is an old idea, right? Just by looking at the distance of 5.7 versus 4.3, telling about reactivity, shouldn't conformational dynamics still play a role?
That's not only conformational dynamics, it's sort of a electronic interaction, coupling.
Electronic interaction.
Okay, which I will maybe towards the end I will be able to allude to a little bit.
But the distance might be dependent on the structure of that.
Yeah. So you cannot exactly match that structure. So it's sort of a dynamic orientation effect, and all those things also comes in the picture. So we actually, by doing all these studies, we showed that this is the heme in case of HRP, having histidine as the axial ligand, and there are several charged residues around it. And we showed that there is a sort of a very strong interaction, salt bridge interaction around the heme that stabilizes the heme so that it doesn't get oxidized by hydrogen peroxide. So if you take myoglobin, add hydrogen peroxide to it, myoglobin itself will get destroyed. Hydrogen peroxide is strongly oxidizing. So if you add it in your hand will burn. Okay.
One thing I want to ask, this 42 or whatever.
Yeah.
So the tail is neutral, is it? So they don't really bond with iron, or it's always the ring part goes.
The tail part doesn't have. See, you have to have the nitrogen which has lone pair of electron. That's what they call it ligand.
So that's the thing.
I'm not going into those details thing. Carbon cannot act as a ligand unless there are special cases. Those are all carbon. Histidine, actually, I should have shown 2 nitrogen presents, but yeah. Okay, now let me come to this last part, where is cytochrome P450, which has taken my almost 2 decades of research career, and maybe half a dozen or more than that PhD students have done their PhD. This is a very, very important and interesting enzyme which does the synthesis in our body. So it's a synthetic wizard. So it can make steroid synthesis. As you know, steroids are molecules having highly stereospecific orientations. Okay, these are hydroxyl groups, and nature synthesizes them from cholesterol. Although cholesterol is bad for us, but without cholesterol also we will not grow up, because cholesterol makes all those steroid hormones. Okay, so and that is done by these P450 enzymes. Okay, it also does another important job is to drug metabolism, or any type of xenobiotic toxic things which are not soluble in water. So anything which is soluble in water goes away because water is the major solvent. But those which are not soluble in water or sparingly soluble, they get into the cell and can actually cause mutations or some disease and things of that type. So P450 enzymes are the ones which actually inserts oxygen into them, so make them polar so that they become soluble in water or they get fragmented. And that's the chemistry happens in the drug metabolism. People have been using this enzyme, trying to use this enzyme to do synthesis, okay? Because this is the one which can do stereospecific synthesis. So these two gentlemen are sort of given the credit to discover this. They discovered in bacterial source, and they found that this is the enzyme which actually found in bacteria, in plant, and their color is red, by the way. This protein also has a color as like hemoglobin and myoglobin, but we don't get to see that color. Even in milk has this, okay? And this is more ubiquitous than hemoglobin, myoglobin. So we did a lot of studies. I don't want to go into detail of that. For example, we looked at thermodynamics of how the protein structure, on binding of substrate, whether stability and all those things. We also found very subtle conformational effects. This was the work of Somen's PhD thesis, where there is a small conformation change in a threonine residue actually helps in stabilizing and also binding the substrate inside the protein near the heme center. Okay, so let me not go. And he also, Murugan converted the axial ligand in P450 is a cysteine, which is sulfur. So he changed that to methionine and histidine, and so that you can actually vary the properties, but just by changing the axial ligand. We have been trying to change this property of this enzyme by doing mutation. One of our initial work where we could convert indole to indigo blue, which was the primitive blue colors, blue genes color and all that. Wild type protein, that unmodified one, does not have that catalytic activity. We mutate it, it shows the color. That means you can actually introduce a new type of function to the enzyme by doing this design thing. We had actually, DBT liked our work on, this was a DBT funded project where we could show that gamaxine as well as dieldrin, which are used extensively as a pesticide, they can be destroyed by using this mutant of P450. Long story short, we realized that this enzyme, this can do the catalysis very well, but it is not extremely stable. It's a mesophilic protein. So because of that, it could not be taken further. And then we found that this P450s actually form a superfamily. That means there are a large number of P450s. In our body itself, there are different P450s present. Those which are doing steroid synthesis are different class. Those which are doing drug metabolism are different class. They will not do, their functions cannot vary in mixing. In plants, there are bacteria also there. Okay.
They also link to allergy?
No. They're just proteins. Proteins will not link to that. So I have shown few structures. They have names are very interesting. I don't want to go into detail. And they have basic structural features are very similar. Okay. On the other hand, and the general reaction they catalyze are also same. Where RH is the substrate molecule, which is generally a hydrophobic molecule, maybe hydrocarbon, maybe a fatty acid, okay, or some organic dye or long chain petroleum product or something like that, plastic and things of that type. And you can actually, and you have a heme. On the top of heme, you have the place where the substrate binds, and then substrate gets converted. It forms a mostly hydroxylated product or epoxylated product. Francis Arnold actually worked her whole life on the P450 BM3, which is Bacillus megaterium, is a soil bacteria. It is also not thermostable, but she has shown it can do wonderful catalytic reactions. So this diversity of P450, that has actually made people think that, okay, we can use the one P450 to do one type of function, another P450 for another type of function and things of that type. But some of those very useful P450s have been found to have, maybe it is found in some pathogenic bacteria and things of that type. So came up the idea that we can re-engineer a known P450 enzyme, Cytochrome P450 enzyme, and use it for the type of function which you want. And Francis Arnold used the directed evolution method, where she mutated this many amino acids randomly and found out whether it can catalyze certain reaction, screen it, they call it. And we tried to think whether we can do this rationally, that means identify amino acids which are sort of identified based on in silico analysis of the structure, and then find out whether we can get this or not. So that was the type of things. And we then concentrated on, this was the time my interaction with Japan started. And we found that in Japan, in hot spring, a bacteria called Thermus thermophilus, which has a P450, which is termed as CYP175A1. And this P450 is extremely stable. Its midpoint temperature is 88 degrees. That means even at 100 degrees, it is not completely destroyed. Okay. And later on, there are other P450, like this sulfur bacteria has CYP119, which has even more stability. But we stick to this CYP175A1, which is extremely thermostable. So you don't really need a refrigerator to work with proteins. Okay. And we then used this technique, which is a sort of a routine technique for us now, that we do a protein engineering around the heme center and change the activity, the molecule to which it's reacting, and by which you can actually, this monoenoic fatty acid, which is, there are unsaturated fatty acids like, olive oil, mustard oil, and things of that type. They are very good because they can be used for making, what they call omega-3 fatty acids and all that. So we try to use them and see whether we can convert them into, hydroxylated fatty acids and things of that type. And also try to make this aromatic hydroxylation, and which resulted in a very nice dye, which I don't have time to talk to you much. Basic idea is that we looked at the mechanism of the enzyme, looked at the structure of the, this is the heme, and surrounding of the heme is the one which is our point. The way, the directed evolution works is that anywhere of the protein, the modification can happen. Some of them may not be relevant. But what we are doing, we are targeting certain hotspots and modifying them. And that's how it is much more directed in this. And these are some of my students. I have not listed the postdocs who have been, concentrating on various type of activities. Some of them are natural functions. For example, we can have either formation of hydroxylated product or epoxides. Okay, these are being either used as sort of nutrients or in the drug synthesis and things of that type. And also there are lots of unnatural function, which generally they call it abiological function, where you can have a, for example, cyclopropanation, which is CC coupling. You can also have a SIH insertion, all other things which has synthetic chemistry applications. So you can, you have a very wide, landscape for the study of the chemistry using this large macromolecular system. So now you don't talk it protein like a biological molecule, rather than it's a protein, a polymer, which is a ligand, and producing a environment around the metal center, because actually it is the catalyst is the heme. Heme is the catalyst, and the surrounding environment are the ones which are very, in a subtle way, is changing the property, changing the way the reaction goes on. So we do molecular modeling, and docking is in silico analysis of the protein structure to find out what might be the type of structure and what type of orientation the molecule might take it. Okay. Here we have a very fruitful collaboration with a very young colleague, Dr. Satyesh Dubey from Sibnadar. So he specializes on molecular dynamics of heme proteins. Mostly he did his PhD with Sasson Sheikh. So they have been working on P450s. So what we identified that this is the P450 catalytic reaction. And here what happens, RH is a substrate. It can be a small molecule, it can be a large molecule, and it has to go inside the protein. So if it has to go inside, means there has to be some sort of channel through which from outside it has to go in, and there has to be a side where it has to come and bind. So there is a substrate access, there has to be substrate binding, and also the byproduct is water. So there has to be a channel through which water has to go out. So these three parameters has to be optimized. So it's not possible manually doing it. So there is a sort of a scheme evolved where we do molecular docking followed by inspection, the lowest energy pose, then identify the hotspot, and then we do the in silico mutation. That means on the structure we make the mutation, and then again do the molecular dynamic simulation, do QMMM calculation so that you can actually optimize the energy and other things. And that's how finally you validate a structure which is predicted by the theory. So this is a very extensive work. I'm not an expert of the theory part, of course, which was done by Satres. And this has resulted us to do something really wonderful. We could demonstrate that a single mutation can actually change the orientation of the substrate, which is something very nicely demonstrated. And the way it is oriented near the metal center, that will decide exactly where the hydroxylation will happen, whether it is going to be omega-3 fatty acid or it is going to make a hydroxylation somewhere else. Okay, so these are very, very important in terms of this product formation. Tahir has done this, he has identified several hotspots. Some of them are for substrate binding, which are, for example, these two. And then water channel is this L80 and V220. And the entry channel are these two. So then what is done is we made a library, very small library, where you can actually show that the activity increases several folds, almost 20 folds, by making this type of things without actually this rational thing. I'm running out. So I think this I will not talk to you about. These are the fatty acid binding and things of that type, where we, Shivdas had shown how nicely we can make a dye called Rasix blue, and studied the mechanism and different type of color compounds. I will just take 2 minutes to tell you about something which we have ventured into. It's called abiological catalysis. This was actually became a hot thing when John Hartwig published a paper in Nature in 2016, where he showed in case of myoglobin, if you change the heme with different metal containing porphyrin, and finally he showed that it can catalyze the reaction where these are called carbene source, where diazoacetic acid or analogs, they can actually form CC bonds and putting a CC or CO bonds and forming these type of products. Okay, and in protein they can be highly oriented type of product, and that has in fact before he published, Francis Arnold actually showed that you can catalyze this type of reaction exactly same. You can have this diazoacetic acid and acrylamide, and you can make this milanacipran. He's a actually antidepressant drug. And when you do that using this P450 catalysis, you and what she did, she used this evolution by the directed evolution and could get yield as much as 81%, and these are very highly oriented, and could get the enantiometric excess of 42%. And further, she showed that you can do what is called site saturation mutagenesis on those and get 92% enantiometric excess. So you can actually use this enzyme to make an antidepressant so precisely and much cheaper in that sense. But of course, her enzyme is water soluble, it's not stable and things of that type. So we have been trying to use this for getting, this type of, value-added products by modifying the protein active sites. Abhijit and Vijay modified sperm whale myoglobin to catalyze this type of reaction. Now question is that whether we cannot recycle them, so we can make a heterogeneous catalyst. So Abhijit actually made a metal organic framework as a solid catalyst. Inside that, you put this mutant protein so that it can catalyze this reaction. So that was quite good. It could be recycled several times, at least 3 times. And it also works at very low pH. But this is something which is very exciting for us, not exactly using the heme, but this is a Japanese collaboration I had with Taher as the key person. We took ferritin, and ferritin is a very, very stable protein which is found in bacteria as well as us. Natural function is to store iron, and it forms a cage type of thing. And it is also thermostable. And you can make, so natural function of ferritin is to store iron in the cavity. So it has around 8 nanometer cavity. Overall size is around 12 nanometer. So you can make ferritin without iron, and then you can load any molecule of your choice. In this particular work, we loaded iridium complex to catalyze stereospecifically transfer hydrogenation reaction. Okay, but this has given us some idea. He had shown how exactly to increase the loading of iridium and things of that type. That demonstrated that we can actually generate the mutant, which is actually my next part of my future plan. So beyond TIFR, is to make heme sitting inside ferritin and doing this catalysis. And it's also very, very stable. So I don't want to go into detail. He has done lots of things. Kinetically, it's very fast. Now, the last slide is, we have this plan of doing this CC coupling using this, synthesizing this antidepressant or many other type of drugs. Using one of them is silica base, where you can covalently link heme and catalyze. And JD, Deepa, and Vivek are guiding us on that. And also this using the MOF metal organic framework or the covalent organic framework, we can make this cage structure where heme can be put in, or heme analog. And Sai has already made some of those type of things. And as I told you, ferritin cage also can act as a host for this. This all work which I presented is, I couldn't cover everybody's, of course, work. And summary is more or less, which already has told. I have told that small iron complex heme bound to various proteins and enzymes and carries out several type of functions like oxygen transport, storage, electron transfer, all these things. And we have done a lot of biomimetic studies using micelles and other things. And we could show that redox potential depends on the environment, and you can change the redox potential in a protein by making mutation at a non-bonded amino acids. And also we could introduce peroxidase activity in non-peroxidase enzymes, proteins. I talked to you about rational design of the active site in P450, thermostable P450. And last thing is we are having efforts to develop heme containing. So you don't need a protein actually. You can actually design this anisotropic environment by doing some chemistry in a nano cage, which can act as a catalyst, which can be recycled for infinite time. So that's the take home. And these are some of my people who have done PhD or postdoc in my lab. Currently, Atish is trying to finish it off, his PhD, and Dinesh is a joint student with Bombay University, so he will take some more time. And Dr. Takafumi Ueno is my collaborator in Tokyo Tech, where Tokyo Tech has invited me as a specially appointed professor, so I will be there for some more time. And collaboration with Satres, I think will go for some more time. And we also had some interaction with Debabrata Maiti. I'm very proud that one of my postdocs, Dr. Nusrat Sangamitra, she has been exception in the sense that she has started her own company called Cygenica, and they have recently got a US FDI approval for doing the initial studies on the drug assessment and things like that. She has got this President Award and many others. She has been doing recently. She had some this CNBC that what they call it, Shark Tank, where she had to make presentation. She got some $3.5 billion. And of course, I thank you for your sorry.
Thank you, Shyamalava. Thank you for the very descriptive story of lots of very exciting moments in the career. We'll take questions, maybe a couple of questions, and then we can all walk down together for a little bit of coffee. Oh, you're on your own.
Yeah. Yeah. Hi, Shamal. Yeah. So, some trivia actually. So the heme, I mean, heme seems to prefer alpha helical folds, right? I mean, like, is there any particular reason?
Not necessary. There are most of the globular systems where heme is present is, alpha helical, but there are, for example, there is a protein called QA, where it's not really helical thing. But yeah, most of the heme containing proteins are alpha helical. But not all of them. Yeah, it prefers, but I don't know why it is.
Can I follow up another? Or?
Yeah. You can ask another, then Shudipto.
Yeah, just a general question about. Just a general question about protein stability. You said stability was a major factor. So, but what do you mean by stability? Because you just need a protein solution which is operating under some ambient conditions, right? I mean. Is it stability because you have to maintain very specific solution conditions or a particular temperature range?
See, for us, stability is important because we would like to make it as an in vitro catalyst so that it should work at room temperature. But in vivo, inside the cell and things of that type, maybe stability has some stability is a measure of interactions, how exactly the metal center is interacting with the protein matrix. And we see that, for example, the heme has this propionic acid side chains. So they do interact with amino acids, and if some of the interactions are destroyed, then it is no more stable. So that type of, you can map the stability of a heme domain in a protein in that way.
Okay.
Yeah. So beautiful summary of all the very nice work over decades. But overall, if I ask a general question, so there are three types of approaches that people are taking for designing proteins. One is what is mostly followed by you, which is a rational design, looking at the structure, thinking about it, and doing the mutations around. Another is Francis Arnold type thing, where you do directed evolution, where you make the bacteria think about it and do. The third, of course, is the artificial intelligence-driven thing that Google and David Baker kind of people. So each will have their own pluses and minuses. Would you like to comment on this?
Yeah, I think, nature has evolved, okay? So, and of course, nature has one limitation is that you are using amino acids, which are real amino acids, okay? So this directed evolution as well as this rational design, what we are talking about, has this limitation that you can, it's not easy to change the, space into using artificial amino acids. But when you are doing, synthetic modeling, where, you can use different type of other polymers or, you can have that, you can achieve that better. But, ultimate thing is that the purpose is you want to make them viable as a, in the long run as an industrial catalyst. And for that, the cost effectiveness. So the directed evolution, it takes, much more effort for, screening. On the other hand, rational design is a very, well-defined and few number of modifications. But the efficiency wise, it might not be as much as in terms of the directed evolution. And the other approach is expensive in terms of the making the catalyst. So that's somewhere you have to compromise in terms of its.
Can I ask?
Yes, yes.
Is it on? Okay. Yeah.
I really enjoyed, although I don't know the things much, but I was curious to know this, this iron valencies that actually decides, how actively it will bind with other agents and so on. So it can bind with oxygen or say toxic things and so on. So I was just curious to know, did you really try to manipulate valency when, for example, it is bound to say carbon monoxide, so which is toxic, so that it unbinds it? Is it like some enzyme or something which could really changes the configuration or valency in some way, so that the toxicity can be relieved? You can.
Okay.
So something of that sort.
If we really look at the, if you want to understand the valency, it is oxidation state.
Yeah.
Okay.
So,
And oxidation state is very closely connected to the redox potential, oxidation.
Yeah.
So if a redox potential of, let's say, iron in a heme is X, at which it binds certain things.
Yeah.
Now, if we can change the redox potential, then only it can change the chemistry. Changing redox potential is not very easy because, it depends on, that's not the whole
but
part
you
of the
have
thing
done
is
in
that
some
you have to
cases
change
like,
the environment.
but you have done in some cases
Yeah,
like it has gone to say 3
Definitely.
or 4 also.
Yeah. So that is what is done. But carbon monoxide poisoning cannot be get rid of because it's the same chemistry, same redox potential of the metal, which is important for oxygen as well as carbon monoxide. So you cannot discriminate between oxygen and carbon monoxide. That's the problem.
I see. So, okay.
Otherwise, for example, you can change the redox potential for a metal center for converting it into doing some other type of function as a sort of. But if there are two competing things which have the same redox potential affinity, there you cannot discriminate. Like carbon monoxide and oxygen needs ferrous.
But you can manipulate the valencies and so on.
You
by
can
changing the
manipulate, but
functional groups
oxygen
and-
binding affinity will go down if you make it ferric. That's the problem.
Yeah. I actually, if the metal ion oxidation potential is little bit different, then the lability of the ligand can be modulated. So
Sorry?
The lability of the ligand can be modulated. Iron 3 never will bind CO.
Yeah, but
if you can make iron 3,
but
the
you
redox
can make
potential.
iron 3,
Right.
but then iron 3 will not bind oxygen also.
Anyway, yeah.
Then
That's
purpose
the
is
problem.
not.
The
The purpose is
purpose
difficult.
To make it something,
Yeah.
so that-
Yeah.
Okay. We have no questions, so let's actually walk with Shyamalava towards our nice destination of having some coffee and a little bit of snacks. So thank you all for coming.